At a very high level, what we're looking at is microbial communities in nature and also microbial communities on humans. And so in both cases, we refer to those as a microbiome. So every human has their own individual microbiome, but we also have microorganisms everywhere on the planet. And these microorganisms carry out important processes for nutrient cycling, carbon cycle, and they're also, for our own health, they're of course extremely important in digesting our food, protecting us from pathogens and disease. We have had amazing aha moments. And just recently, we've developed a better way to assemble DNA sequences from soil. This has been a challenge because soil is the most diverse habitat on Earth, much more diverse than the human gut. One example is frozen soil in the Arctic, and this is permafrost. And we know that permafrost is a huge reservoir of carbon on the planet. It's basically frozen carbon since the last ice age. It's about as much carbon in permafrost as is currently in the atmosphere and in plants on our planet. But it's frozen. So what happens as the climate warms and the permafrost starts to melt? We know that there are microorganisms that are in permafrost. So as the permafrost melts, those microorganisms can become more active because it's warmer. Then they can in turn start to degrade that carbon, that organic carbon that has been trapped for thousands of years in permafrost. When they do that, they can also release carbon dioxide and methane that are very potent greenhouse gases into the atmosphere. We have more microbial DNA in and on our bodies than human DNA. So we're 10 times more microbe than human if you look at the number of cells on our bodies. There are some real strong correlations to certain microbial compositions and some kind of diseases, as well as diet. So this is relatively new knowledge within the last decade or so. Once that knowledge has become available, the next step is to think, well, how can we take that a step further and go towards more personalized diets or precision medicine treatments? Because the composition of microorganisms can also influence how drugs are metabolized in our bodies. And so as we get more information about our own individual microbes, then we can tailor our drug therapies or our diets correspondingly. We're dealing with complex systems. The microbiome is still incredibly complex. We're thinking about thousands of microorganisms, each carrying out different processes. In order to understand what they're doing at a mechanistic level, we need to have these very detailed types of analytical capabilities, such as we have at PNNL. The data that is generated is enormous. We get millions of genes and information about hundreds of thousands of species. And so in order to be able to get a better picture of this puzzle of interactions, we need supercomputing facilities. At PNNL, we have the whole package, the analytics and the computation. So my vision would be to be able to go beyond the current state of the art, to be able to eventually establish a microbiome center of excellence here at PNNL so that we can take advantage of our technical capabilities and our expertise in microbiome science